As we start talking about the technical details of how we extract energy from our food, we're going to start with glycolysis, which is basically taking a glucose molecule and splitting it in half over several steps to produce a little ATP for us. Now we know we can use more than just glucose for food. There are fats and proteins and starches as well, but this is a good place to begin the journey of understanding chemically what goes on inside of a cell. And it's a pretty universal process. Animals can do it, bacteria can do it, plants can do it, fungi can do it. It happens out in the cytoplasm and is really the step before the real payoff that happens in the mitochondria. Let's take a look. We start with sugar for a good reason, because it's a pretty readily available energy source. And really all carbohydrate roads lead to glucose. Plus, there's this other matter of convenience. So here I put some starch into water. You can see when I stir that up, it's all cloudy. If you've ever done this yourself, you know it's all going to settle back down to the bottom. Obviously, not something we want in our bloodstream. It'd be difficult to transport it around that way. Now, unlike that, we have sugar. I dumped the same amount of sugar in here earlier. You can see that sugar's swirled up in there, but then dissolves pretty readily, which makes it nice for transporting around in our bloodstream, or even plants in the water that flows up and down their stems. So that's kind of one reason. Plus we can pretty easily break down starch into glucose. So that's kind of where we begin with glucose. And so looking at this on an energy diagram, this is kind of a summary of glycolysis. Very often we really simplify the chemistry, so we're not trying to look at these big molecules and unnecessary stuff. A lot of times we'll just follow the carbon chain. So here I have 6 little dots representing the 6 carbons of glucose, and we end up with 2 different pyruvate molecules, each with 3 carbons. So that's kind of what I meant by we break glucose in half. And that happens over a bunch of different steps. And we start with glucose and we actually put energy in. We add ATP to this reaction. So energy goes in for the first several steps, and then energy is going to come out in the other steps. And out of this we just get a couple of ATP, some other electrons, and of course a lot of energy is lost as heat. We have to obey the laws of thermodynamics, which say we can't transfer every last little bit of energy, so we do lose some energy as heat as well. And these pyruvates are not the end of the story. They still have energy in those chemical bonds, and we have more energy we can extract from that, but that's going to have to wait till later. Well, here's a pretty thorough summary of glycolysis. It's intimidating, I know. It's a lot to take in. So we start over here with glucose. Let me point out a few things just to help you make sense of it. And you should do this when you look at any complicated diagram. First, we have several molecules highlighted for us here in yellow, ATP. Obviously that's important. So is this one over here on the right, NADH. So they're given that nice bright color for a reason. Kind of related to those are these phosphate groups, those PO4 groups, so phosphorus and oxygens. Transferring those around is how we're going to make ATP. Remember, ATP is an adenosine with 3 phosphates, so obviously those phosphates play a key role. So that's another one. And then lastly, each of these steps, labeled 1 through 10, is a chemical reaction that is facilitated by an enzyme. So this means there are 10 different enzymes involved here. That's a pretty complicated set of reactions. Now, I've not named every enzyme or every molecule along the way. We just start with glucose, and let's pull out some of the major ideas here. So I said at first we add energy. So we're going to add ATP, which transfers a phosphate onto that group. We add even more ATP, which adds another phosphate over here. In step 4, we go from a 6-carbon sugar to a couple of 3-carbon molecules, this guy and this one. This one at the top gets converted into the same thing, glyceraldehyde 3-phosphate. Not important that you know the name, just gets converted to the same thing. So we end up with 2 of everything from there on out. So you'll notice the process doubles, but rather than write them twice, I just added that there's 2 of those and 2 of those. And in the end, we end up with 2 pyruvate molecules. So everything gets doubled. Well, this step we get a little bit of this electron carrier, NADH, full of electrons. We'll kind of put that one off till later. And in step 7 here, we get 2 ATP out of that step. All right, finally some payoff. And then in this step, we get 2 ATP out of that one. So in summary, net, we get 2 ATP. We used 2 up here, so that was a -2, and we gained 4 down below. So overall, I'm 2 ATPs in the positive. Got a little bit of energy that I can use out of it. Well, there's glycolysis. So what comes next? Well, what do we have now? We've got a couple ATPs. Obviously, those are going to go somewhere and help the cell do work, whether that's pumping ions around or moving muscles. It's there to do work. We have a couple of these weird electron carriers, NADH. We'll deal with those later. We can give them to the mitochondria, and they'll help us make more ATP from that energy. We also have 2 pyruvate. Now what happens to pyruvate next, because there's more energy stored there, kind of depends on the circumstances in the cell. Let's take a look at that. So here is a much briefer, more digestible version of glycolysis, kind of telling you the energy input, half of it, and the energy output, half of things, and that we end up with these 2 pyruvates. So what do we do with those pyruvates? If we have oxygen, and of course we have mitochondria, then we can keep breaking down that pyruvate and make up to 34 more ATP. Wow, 34? Are you kidding me? Yeah, 34. That's a big payoff. If we don't have oxygen or we don't have mitochondria, then we do fermentation, and that's the end of the line. We've gotten 2 ATP out of glycolysis, and that's all we're going to get for every glucose that we put in. So obviously it's better to do this step, but that depends on whether or not you have oxygen and whether or not you have mitochondria. And different organisms vary in their ability to do this. As far as we go, obviously this is our preferred route. It gives us the most ATP for the energy we take in. So most of the cells in the body are going to do that. But some of them do this, namely our red blood cells, for reasons we'll go into later, but also some of our muscles, especially those heavy lifting muscles that don't really have the time to go through the extra steps up here to get that big ATP payoff. They just settle for fermentation. They're inefficient, but they are powerful. So that's glycolysis, a complicated first step in the energy extraction process, but it's a necessary one. And it's an important one for us to build on. A lot of the other ones are going to have some similar themes that we'll keep looking at in future videos. That's it for this time. Until next time, keep appreciating life.